The kit used for DNA extraction is Kiagene DNA mini kit, which contains ETL, ALAE, proteinase kinase, and alcohol. The first step is to isolate one live micro-arthropod specimen from a colony using microscope and transfer into Eppendorf tube containing 20 micron of ATL buffer. The second step is to crush the micro-arthropod specimen. Followed by addition of a further 160 micron of ATL buffer and 20 micron of proteinase kinase. And mixed using a vortex and mini centrifuge for about 15 seconds. Again, quick mixed with the vortex, and the sample is then incubated overnight at 56. Most of the time, a gelatinous layer created in the mixture after the incubation. The third step is to add 200 micron of AL buffer and then mixed using vortex and incubated for 10 minutes at 56, followed by addition of 200 micron of 100% of ethanol and mixed again. The fourth step is to transfer the solution to a mini spin column. Followed by the centrifugation for 1 minute at 8,000 RPM. The fifth step is to place the column into a new tube while the previous tube containing the flow-through is discarded, and 500 micron of AW1 buffer is added to the column. Followed by centrifugation at 8,000 RPM for 1 minute. The sixth step is to transfer the column into another tube again while the previous tube containing the flow-through is discarded, and 500 micron of AW2 buffer is added to the column. Followed by centrifugation for 3 minutes at 13,000 RPM. The seventh step is to transfer the column into another tube again while the previous tube containing the flow-through is discarded, and 50 micron of AA buffer is added to the column. followed by incubation for 1 minute at room temperature, and centrifugation for 1 minute at 8,000 RPM. The final step is to repeat step 7, but with addition of 100 micron of AE buffer. Followed by centrifugation for 1 minute at 8,000 RPM. The sample containing extracted DNA is stored in the freezer at -20 until utilized for the molecular genetic studies.